All right, good morning. My name is Dan Scher. I'm a biochemical geneticist at the University of Alabama at Birmingham, and I will be moderating this session. It is a pleasure and a privilege to introduce my friend and colleague, Dr. Bob Kesterson, who is a professor of genetics at UAB. Bob runs our transgenic mouse core, and he is going to be, whoops, I lost my notes here. This is what I get for, okay. The title of Bob's talk will be CRISPRs and Gene Editing, Laboratory Use and Clinical Future. Bob, take it away.
How about now? A little loud? How about now? We good? In the back? Great. So I'd really like to thank Dan and the other organizers for inviting me to talk at this meeting, because I think it's an opportune time to start introducing and maybe talking about my title today, the subject. So CRISPR technology, many of you have heard about CRISPRs, and some of you probably are wondering what's all the big fuss about, and so I'm going to tell you a little. So I've been in the mouse business for the most part for the last 25 years or so. My research early on at Baylor got me interested in making simple transgenics, and then we also learned some technology from leaders in the field on how to manipulate the mouse genome. And the mouse became the premier genetic tool for model organisms, primarily because of the unique ability to manipulate embryonic stem cells in cell culture, introduce a mutation for any gene that you wanted for the most part, and then reintroduce those cells into an animal, and then make a chimeric mouse and breed these until you got germline transmission of that mutant allele. And that's been a real boon to learning more about gene structure and gene function, and so it's often called reverse genetics, where we start with the gene and then figure out the phenotype later by manipulating it. But this has taken a long time, and so we were kind of frustrated, and you know, we've been wanting to branch out. We're limited to the mouse, and so it's taking too long, and there's a worldwide effort to create a knockout of every mouse gene, and we're now about 10, 12 years into that effort. We've knocked out about 6,000 or 7,000. And so it was just taking too long. But then the skies recently opened up, and what was delivered to us was the CRISPR-Cas9 system. And I give credit to Tyler Jacks. I stole this concept for the slide. Meeting in Orlando, genetics meeting last summer. And really what I'm going to do is tell you about the CRISPR-Cas system and really outline, tell you a little bit about the history of it, how we're using it in a laboratory, tell you about some remarkable things that are being done. That it's, you know, you open and every week seems to be a new application for CRISPR-Cas9 and doing genetic and epigenetic regulation. There are some concerns about gene drives, and this is the concept of manipulating an organism and releasing it into the wild such that you persistently pass on a mutated allele. And I'll start telling you about clinical trials and the future applications, because I think for many of the people at this meeting, it is, you know, what are we going to do clinically, and how is this going to help your patients? So the simple introduction to the CRISPR, so CRISPR stands for Clustered Regulatory Interspace Short Palindromic Repeats. Clearly, CRISPR is much easier to say than this long title, but what this is that bacteria have their own immune system. And the immune system that the bacteria have is that after initial infection from a phage of some sort, then the DNA or RNA from that phage is copied, and it's inserted, short fragments of it, into a locus in the bacteria genome. And there are these proteins called Cas9 genes, and in this case, the model system that you mostly hear about is Cas9, and that's from Streptococcus pyogenes. And so the Cas9 protein, what happens is these bits of bacteria DNA, when the phage is reinfected, what they do is they mobilize, they copy these short stretches of DNA, and actually they make an RNA, so they make this copy RNA that's uniquely represents or recognizes the bacteriophage material. And then along with a tracer RNA that works with the Cas9 protein, this puts this into a certain configuration that allows for specific cutting of those nucleic acids from the invading species. And so this is a memory, so it's an adaptive immune system that bacteria have. And so here's a short timeline of where we are with understanding. I mean, the first papers that were published were in the late 80s, and then the idea of a CRISPR, these short regulatory repeats, was really recognized around 2002, and start unraveling what the function of these particular genes are. And around 2012, it was really the adaptation of this system to manipulate mammalian cells that really has changed the way we're doing things. And in recognition of this, here are the major players in the field. The Breakthrough Award prizes were given to Dauda and Charpentier. And so these were the first two that were recognized with these early papers. But it was really Feng Zhang at MIT that then started adapting and manipulating mammalian genomes using the CRISPR-Cas system. And in fact, you've probably read a lot about the patent wars that are going on. These are the three major players in the major institutions. And the first patent has been awarded to MIT and to Feng's group. So another way of looking at the history of this is, like I said, everything accelerated around 2012. Feng's group applied for the patents. He published the first paper on editing human and mouse cells. So this is the first demonstration of the mammalian cells being able to edit using this. Quickly followed were people engineering zebrafish, and then moved on to plants, other species, macaques, primates, pigs. So the key here, and I'll show you in just a second, is that these are very easy to work with. And so now we even have the first CRISPR that was used to edit the human genome was around 2015 by a Chinese group. There's since been a quelching of that worldwide. And so I don't think that until we solve some other issues, there's certainly not going to be any germline type manipulations that would then introduce mutations into the human germline. So why is this technology so important? And what makes it ideal for modifying the genome is the fact that it's highly efficient at cleaving DNA. So it's high potency, and it's highly specific too. So we can engineer those short guide RNAs to any sequence for the most part. There are some limitations for each Cas9 or each Cas-like protein, and I'll show you those in a few seconds. And there's broad applications both in vivo and in vitro, and probably the thing that makes it the simplest to use is that it is, anybody who has basic molecular biology skills can use this. So I equate the CRISPR-Cas to really what happened with PCR. So this is now a tool that's going to be rapidly adapted by every laboratory doing research, and mainly because of the speed and ease of the use. And then, of course, the potential applications are that we could possibly cure some diseases with this. We can introduce foreign genes. The other thing is the Cas9 system allows you to not just edit one gene at a time, but there's been demonstrations that you can modify multiple genes in the same cell at the same time. So in fact, there's been animals made where 5 or 6 different CRISPRs were injected into a single embryo, and you knocked out 5 genes at a time. So remarkable pace at which you could manipulate the genome. And then from a regulatory region, I think this is where NIH has struggled, and your biosafety committees are likely struggling at your own institution, is, well, we're editing the genome, but we're not leaving any residual sequences behind. So this really isn't a transgenic animal that you're making. I mean, you're simply cutting DNA in a certain region and allowing repair mechanisms to then introduce a mutation. So let me go on and show you how this works a little more. And so targeting by gene editing using nucleases is really nothing new. So we've known about talons and zinc fingers. We've had those for many years now. And as I said, the key here is that The CRISPRs are really easy to make. And in fact, now you can purchase purified protein. You can order the oligos that you need for the trace, for the guide RNAs to target your gene of interest. You can order it from numerous oligonucleotide synthesis companies, and you can have these materials in a matter of a week to then manipulate any gene in a cell line or in an organism. So the zinc fingers, the talons, and the CRISPRs, they all have the same thing in common. They're simply nucleases. They just introduce a double-strand break in DNA. But the difference is, as I said, it's easy to make the CRISPRs, and so the cuts are much easier to make, and you can tune where you want to cut based on synthesizing these guide RNAs that then interact with the Cas9 and the tracer RNA, which has already been packaged into a single unit. In fact, now, when you order your sequence that you want to target, and there's an online website, Feng Zhang's group at MIT maintains a program where you upload the sequence that you're interested in targeting. It'll search and give you optimal guides that will cut the DNA of interest in a particular region, but then it also scans for potential off-target sites. So what is that sequence represented anywhere else in the genome? And it'll select the ones that will most likely work with the highest efficiency and not target another region. So the key here with these nucleases is that one, they make the double-strand break, so DNA doesn't like, in a cell, doesn't like to be broken, and so there are repair mechanisms for that. And the non-homologous end joining is the most efficient way of repairing that DNA. And during this process of non-homologous end joining, the polymerases just kind of jam things back together. They grab two pieces of DNA, they sew them back together, and in that process you're inserting or deleting a few bases at a time, or even larger, there could be large deletions. So this creates indels. So it's easy to create a mutation in a gene, and I'll show you some of the primary data from some of our projects in a few minutes. But the key about this double-strand break is if you introduce a donor template, there's another repair mechanism called homology-driven repair that will use this extraneous bit of DNA that you're co-introducing into the system at the same time of cutting, and it will use homology between your donor template and the regions of the DNA that need to be repaired, and there'll be double crossover events here, and you can then get insertions or corrections. So if you have a long stretch of DNA, like a single-stranded oligonucleotide that can be used, then you can make simple changes like a SNP. So you can introduce a single base pair change or two. You can also use donor templates that have flanking homology arms to do much larger insertions. So for example, you could put in reporter genes, or you could insert a cDNA anywhere in a particular area. So this is the key, is that you can make these modifications if you can efficiently get homology-driven repair. And for the most part, the homology-driven repair is simply a numbers game. If you screen enough animals, the frequency of that homology-driven repair is such that for the small changes, if you're just trying to introduce a SNP and you give an RNA, that happens, depending upon the genetic locus, anywhere from 10% to 60 or 70% of the time. Some people have optimized that for the particular mutation of interest, and so you can get the efficiencies much higher, but that means you can even get biallelic conversion with just a single guide RNA with a very small change. The larger the change you're making, then the lower the frequency. So how are we using this in practice in the lab? So one of the first things we decided to do as we jumped into the system is, well, we wanted to start testing our guide RNAs, and so we decided to pick a few genes that we wanted to assess the ability to do the targeting knockout. And one of the first struggles we had is, well, how do you screen for the mutations? And so we set up a rather simple screen that a lot of people have since adapted. And so if you are introducing a mutation in a double-strand break in a particular region of a gene, if you have primers that are outside that region and you amplify up the piece of DNA, if you have introduced a mutation, then that indel in your PCR products, then you'll have a heteroduplex that forms. So if you don't have any CRISPR activity, if the DNA is not cut, or if everything is the same, your PCR product, you get these homoduplexes. But a heteroduplex will run differently on a polyacrylamide gel than will a homoduplex. So as long as you're looking at controlled DNA, you can screen for the wild type. In this cartoon here, you see that the wild type PCR product may run at a certain molecular weight, but then you see these extraneous bands due to a heteroduplex. So what we do is we test all of our CRISPRs. So we're manipulating mouse embryos for the most part. We're now doing rat embryos as well, or zebrafish embryos. But you inject or you can electroporate in your CRISPR reagents. For the mouse, we can culture these embryos to for about 2.5 days. We'll culture them to the blastocyst stage, isolate these blastocysts, isolate DNA from those, and then run a PCR reaction, and then run this out on a gel and look for these heteroduplex. So that's how we test our CRISPRs. Once we have a CRISPR that works well or has high efficiency, shows cutting activity, then we go back and re-inject into embryos and then transplant these embryos into recipient females, and we end up producing animals that have that mutation. So that's the overall process. Since one of the first genes we decided to target was the tyrosinase gene. And mammals, if you don't make any tyrosinase, then you can't produce dopaquinone and you can't produce these pigment granules that are necessary for melanocytes to produce colors or pigments for the fur or the hair as it's growing. So if you have a tyrosinase knockout, you end up with an albino animal. So we designed CRISPRs to 2 regions of the tyrosinase exon 1 gene. We had 2 different CRISPRs. We just labeled here a 5' CRISPR and a 3' CRISPR, all to exon 1. In our blastocyst assay, then we saw the activity of, so if this is a normal wild-type PCR product, you can see this animal here did not, or here's a control over here, so there's no CRISPR activity. And this blastocyst here, 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 and here, you see these few extra bands. Over here, you see it quite well with this 3' CRISPR. You see extra duplex bands here. So this indicates that these CRISPRs are able to produce extra cuts, and then that's indicating that the CRISPRs are good. If you then take tail DNA, go back and inject these blastocysts and make animals from them, these are founder animals. And so these animals, you can see, for example, a couple here that don't appear to have anything, so the CRISPRs probably didn't work in those embryos. But here's a nice clear extra band, and here's some extra bands. And so in many cases, you see multiple bands that are present here. And we then breed these founder animals out, and we start looking at, well, you know, what do they look like pigmentation-wise? And so what we decided to do is we actually picked animals that were a pure B6 background, and what you can see here is we had one functional tyrosinase allele and one mutant allele. So really all we had to do was inactivate the one allele. And if you look at these animals here, these are founder animals, and what you see is patchiness of coat color. So these animals normally would be black because they had one normal tyrosinase allele, but wherever you see patchiness, you have inactivation of the tyrosinase, and so this shows that these animals are mosaic. And we weren't surprised by that because depending upon when or how soon during development the CRISPR starts cutting the DNA, which cells would then would be inactivated. And what we now know is that we have mosaic animals that can contain up to 5 or more mutant alleles. So we've actually taken a single founder and bred them and had multiple alleles produced from just one founder. So here's these evidence of the CRISPR activity. We also targeted a few genes. A couple of projects we were interested in making for other investigators. The one, a CORT4 receptor gene, we knew that we would get an obese phenotype if we hit MC4, but I'm just showing you the tyrosinase data. And interestingly, we had some animals that were completely white with the mutation, so okay, that means we knocked out that functional tyrosinase gene early on before those cells gave rise to neural crest cells, which then give rise to melanocytes. And then we had some animals that were kind of off-white, and those were the ones that are actually most interesting. Here's from a given founder line. Like I said, you'll see these kinds of tables if you look at papers looking at the alleles for CRISPR-related functions. And so this has kind of been an adopted strategy that most people will report for their different alleles that they're generating. And what you see is that all these are unique mutations introduced by the single round of injections, and you get everything from in-frame deletions to premature stops, and many of these are just simply indels. So this one you'll see that it gained one base pair, but it lost 4 other base pairs. So this one's a -10, a -15. So one of the things we're kind of interested in a couple of these, so the -15 mutation, well, that keeps it in frame. And in fact, we had a -15 mutation. We also had a 3 base pair deletion, which deleted 1 amino acid, kept it in frame, and this produced 2 novel alleles. And because the person who was doing this, Anil Chawla, is from India, and we have lots of tyrosinase alleles with interesting names, so Anil decided to name these 2 novel alleles Chandana and Dusara, which means sandalwood and gray, so these are the Sanskrit names. And we went on to characterize it. So you can see the slightly different pigment levels in these 2 different mutations, with Chandana being nearly complete loss of activity, and the Dusara is a little bit more kind of a grayish color. If you look at the pigmentation in the iris, you can also see the amount of pigmentation in the epithelial layers correlates with the amount of tyrosinase activity that is associated with these mutant alleles. So I think this is one area where the investigators, I've been harping on this in multiple talks, is that don't overlook these hypomorphic alleles, because with these in-frame deletions, I mean, hypomorphic alleles, as geneticists we know, are often the ones that are most informative. You don't completely inactivate activity, but you retain a little function. All right, so there's been a worldwide, so how are we using these CRISPRs? There's a worldwide effort with the International Mouse Knockout Consortium. We've gone through multiple names, so this is now the phenotyping consortium associated with that. And there's been roughly about 8,000 mice that have been knocked out. So 8,000 genes have been completely inactivated so far in mice. So if we have roughly 20,000 genes, we still have a ways to go. And most all of these mutations were made with what's called a knockout first strategy, in which you insert a piece of DNA that inactivates the gene, but also you end up putting LOXP sites flanking critical regions that allow you to convert that knockout allele into something that's a conditional allele. If you look at the number of number of animals in which a gene has been manipulated, around 15% of those are absolutely necessary. So you end up getting embryonic lethality if you have complete loss of function. And so you really need conditional knockouts, which then allow us, using the CRE-LOXP system, if we can flank a gene with 2 LOXP sites and then later introduce CRE recombinase, then we can turn the gene off in a select tissue. And so that's the conditional knockouts. Well, now the Sanger Institute, which is leading this worldwide effort, the U.S. has COMP, which distributes ESLs, and also has several institutes involved in production, including Baylor. They've gone back and modified their alleles to now, or their targeting vectors to now use CRISPRs, because we can now more rapidly create these knockout animals. And so CRISPRs have fundamentally changed how soon we will have knocked out every gene in the mouse. There are numerous efforts worldwide. This is some data from the Czech group. I don't expect anybody to have seen any of this, but this is just a list of projects showing that generating indels, making SNPs, and this is just a summary of some of their data from a meeting early last year, or earlier in the year. And the key to this is that with CRISPRs, they're just so much quicker to make a knockout animal. So if you think about how long it took us to make an animal using embryonic stem cells, typically you'd have to create a targeting vector, introduce that into embryonic stem cells, inject screened multiple clones, identify the ones that had the correct mutation, and then go back and inject those into a blastocyst, make chimeric animals. You'd have to breed these for extensively until you got the mutation to go germline, and then you could cross them back and make double knockouts. Well, if things went well, on average, that process takes you almost a year. With CRISPRs, we can knock out a gene in reality in about 6 weeks' time. It takes you a week to order and design the CRISPRs, do your injections. Actually, this time has been cut to a week from 3 weeks. Microinjected embryos. You can have animals that have multiple knockout alleles as early in that first generation. So I showed you evidence there where we had the tyrosinase, where we've completely inactivated that in that very first generation animals. Normally, you need to breed out those founder animals, identify the mutant allele, and then selectively go back and breed the ones that are clearly loss of function. So very rapid. And then the other issue that is often coming up in any talk concerning CRISPRs is, well, what about the nucleases cutting somewhere they're not supposed to? So these off-target effects. And what I'll say is that of all the projects that have been done by large groups, Jackson Laboratory, Taconic, the Canadian group, the Czech group, the Sanger group, there's been deep sequencing of all those products, and there are virtually no off-target effects in the animals that have been produced. It's a slightly different story in the cell lines, and that's likely due to the method in which you're introducing Cas9 and you're putting it under selective pressure and you're keeping the protein around for a very long time. So those, you do have more off-target effects. So what are we doing with these? Well, and I kind of listed, here's a diagram showing some of the different types of mutations you can make. Insertions and deletions, those are relatively easy to do. Larger deletions are actually fairly easy as well. We've deleted 100 to 200 kilobases for a couple of projects of ours. It's really important. So let's say you're studying two genes that are closely linked to one another, or maybe two genes that have duplicated and then they are, isoforms or related genes. Well, it's very difficult to make individual knockouts, and then if you decide to breed those, it would take a huge number of offspring before you're ever going to get a crossover event. Well, with CRISPR, you can simply target both of them at the same time, or if you have the knockout, you can go back and target the other gene with a CRISPR reagent. So it's relatively easy to make large deletions, make knockouts. The SNPs, so if you're only making one or two base pair change, you can do these with single-stranded oligo repair templates. Those are also moderately easy. Now, not every gene is slightly different, so there's going to be empirical data on this, but I mentioned earlier, on average, people are reporting somewhere between 10 to 60% knockout or conversion with single SNPs. The larger the change you want to make, well, the efficiency goes down. And that shouldn't be surprising. So you're depending upon this homology-driven repair. And so if you want to insert cDNAs, put in reporter genes or something like that, then that's more difficult. LOXP sites are also more difficult. And so if you're trying to flank key exons with two separate LOXP sites, if you think about it, we're going with two different CRISPRs, two different repair templates to try to introduce those LOXP sites. Well, what happens is you'll get one that goes in, but then the other one doesn't go in. Or if it goes in, it'll go in on the opposite allele. So getting two LOXP sites in cis and not in trans, that's a low efficiency proposition. So now what's happening is if you get one LOXP site in, then you simply take those animals and use them, you grow them, expand them, use them as donors, and then insert the second LOXP site. Now, there are also groups that are starting to fully humanize certain genes. So when you hear somebody say humanize, well, that means a lot of different things. For immunology people, it means taking out immune cells and doing adoptive transfer type experiments. For somebody like me, when I say humanize, I tend to find a mutation in the human and make the same mutation in the corresponding mouse gene. But what we'd really like to do is replace the mouse genes with a fully human copy of the gene. And so this is what's being done with back transgenics. So BACMENs, large constructs, are now being inserted and completely replacing the endogenous gene. So then you can start doing your structure function studies or mutations. So that's kind of the history and the background and some early data. We've used this to then branch out. And I'll tell you about a couple of projects that my group has been working on before I get to the clinical utility of these. And so we were interested in a gene called osteocalcin. And the reason for that is it has been reported in a mouse model that this bone protein, so it's produced mainly by osteoblasts, may have a role in not just bone regulation, but also in glucose homeostasis. So it may function somewhat like leptin does, so a signal from adipocytes to other organs. It was proposed that osteocalcin is a signal that comes from bone to talk to other organs. And so we were quite interested in studying this, Now, one of the reasons is the group that had reported all these fascinating phenotypes with their mouse knockout, but there'd been a lot of groups, and there's a lot of controversy in the area because a lot of people haven't been able to reproduce this. Huge number of studies going on in humans trying to assess osteocalcin's function, tying it to adipocytes and glucose homeostasis in humans, and some with very mixed results. And so we were early on, so this is around 2013, we were working with a clinician, a surgeon who was interested in pursuing osteocalcin, and we were going to have to make a complicated animal model. And it turns out that the mouse that all this published data is relying upon had knocked out a region that encodes the mouse osteocalcin genes, but the mice are different than humans in that there's actually been a triplication event around the osteocalcin gene. So the osteocalcin gene and an adjacent adipokine type receptor had been duplicated in the mouse, actually triplicated, and so the mouse knockout had 3 copies of this. So we were going to make a traditional knockout in embryonic stem cells, and this was going to take us, year and a half, 2 years to make this, particularly because we were thinking about just completely humanizing, just sticking in the human gene here. Well, along came CRISPRs, and we decided, well, here's the human gene structure. We don't need to make a mouse, so instead of trying to knock out all the genes in the mouse, which we can now do with CRISPR, why don't we just target a rat? And this is where the CRISPR activity, these nucleases, their ability to work in vivo in embryos, and the fact that you can make them and inject them that have high activity really comes in handy for manipulating other species. And so we made a series of CRISPRs that targeted different exons, and so here's our first injection in rat embryos, and we had a couple of females that had multiple alleles. So the rat gene structure is just like the human. That is, there are only these 4 genes in a row. They're a single osteocalcin gene, so we decided to make a knockout rat. We had one founder rat animal. You can see these complicated patterns up here. So we had one animal that produced 5 different mutant alleles. Here's a summary of some of those alleles. And so we ended up taking F1 animals that had these different mutations, all of which should put the gene out of frame. We had one which is a 312 base pair deletion, a separate line that's a 240 base pair deletion, and a couple of these more complicated ones. And the key here is when we made compound heterozygotes, there was no osteocalcin protein produced whatsoever. So we successfully made a knockout rat model. And then when you look at the functional, what do we find with the phenotype in the rat? Well, they had increased trabecular bone. So you can see here, here's all the hard data, but visually, if you just look at the trabecular bone, you can, from a micro CT scan, you can see the increased bone here. And in fact, if we do a 3-point break assay, if we take femurs from these animals, and then Alan Eberhardt's group did this, in which they increased the mechanical stress applied across the femur, it takes more force to break the femur for a osteocalcin knockout rat. So these animals actually have increased bone function. We looked at the glucose, and we did glucose tolerance tests and insulin tolerance tests. We saw a small, very minor difference. And in fact, these experiments were male animals done at 6 months of age. We've now gone back and looked at males and females at multiple ages, and we always see something like this. So if there's an effect on glucose or homeostasis, it's exceptionally minor. Certainly nothing that mimics what has been reported in the mouse. So we really believe that the mouse model is not an accurate model. In fact, the rat is probably more like humans, and it probably explains all the mixed data that's been generated in the literature, in that original knockout mouse knocked out this adipokine receptor that's adjacent to it, and not just the osteocalcin gene itself. So I won't bore you with the conclusion, but the main thing is these animals have increased trabecular bone, and they really don't have much of a metabolic phenotype. So the other example I will tell you about briefly is that we've had a couple of talks about neurofibromatosis type 1. My chair is Bruce Korf, and he came to me a few years ago about wanting to create some better animal models of NF1. And the reason for that is, here's a review of the disorder. Most of you have heard this several times. The main problem with this is it's not really a rare disorder. 1 in 3,000, I don't consider that rare, although technically by definitions it is. But the problem is you get these malignant tumors, these peripheral nerve sheath tumors, in about 10% of patients, and particularly with patients that have the deep plexiform tumors. The cutaneous tumors don't really give rise to these MPNSTs, but you do end up with these very characteristic phenotypes. And so Bruce came to me and said, well, can we create some better animal models? Because there have been a couple of animal models that have been used by the field for many years, a conditional knockout model and then a couple of different straight knockout alleles. And as expected, if you make an animal that's homozygous for NF1 deletions, it's embryonic lethal, and that's been true in all species so far that's been studied. And so what we wanted to do was start making patient-specific mutations. And we're really interested in making premature stop mutation, and we're also interested in making mutations in, or recapitulating mutations that humans have that are associated with a subclassification of families known as spinal NF. So these are patients that have these deep plexiform tumors that arise along the dorsal root nerves. And in fact, so here you can see this is one of Dr. Korf's patients, and you can see really not much evidence of a dermal tumor. This is a sciatic nerve on both sides. These bilateral sciatic nerves here, you can see the spine, this huge tumor mass of this particular patient. So this is this familial spinal NF. And so we've been interested in there are already many groups that are looking at downstream signaling pathways, and we're trying to figure out, well, can we use mutation-guided therapies to then repair certain mutations? And so we're interested in the spinal NF. We're also interested in the nonsense mutations because there's roughly 20% of the known mutations in the NF1 gene are nonsense alleles. And so it's a large gene, 56 exons, over 3,000 independent mutations have been identified so far. So a huge number of mutations. There's a lot of premature stop mutations. And the reason we're interested in the premature stops is that there is nonsense suppression therapy as a way to change. So normally a premature stop, the release factors recognize this premature stop, and you end up getting truncation of protein, and so you end up with a non-functional protein. The other thing that happens is the mRNA is rapidly degraded. It's recognized as an aberrant mRNA, and so you get nonsense-mediated decay. But there are drugs, the aminoglycosides, that can allow a near-cognate tRNA to introduce an amino acid. And so if it happens to introduce the right one, then you're good. If it introduces an amino acid that leads to, could be a missense, but it could be a functional protein, or it could be a non-functional protein. But the key here is you can get some read-through. And this is not just this is a therapy that's been proven for a few different disorders. It's been tested extensively for multiple diseases. There have been about 100 studies for 40 different diseases. There's been clinical trials. Adalurin is a drug, it's PTC 124 from PTC Therapeutics, that has shown some of the best use clinically. It's worked for Duchenne's muscular dystrophy, so it's approved in Europe. If you have a premature stop mutation and you live in Europe, you can go on PTC 124 and it will alleviate symptoms. So the other thing is we've been making premature stop models at UAB for quite some time. I've created a Hurler syndrome mouse, a cystic fibrosis mouse. So we went ahead and created a nonsense mutation for the NF1 gene, and so we found a recurrent mutation. We introduced this into the mouse, and you'll see this is through conventional ESL transgenics, and I'll come back to the CRISPR in a minute. Our premature termination codon ends up, if we try to make animals that are homozygous for this, you get no neurofibromin protein made, get early lethality around embryonic day 8.5, you have these cardiovascular bleed outs. This is a wild type animal, you get this abnormal pericardial sac that forms. The other thing is the heterozygous animals, if you take and isolate the embryos, you can see that these animals do show nonsense-mediated decay because we only see about 9% of the mutant transcript, where we would expect about 50%. So there is degradation of that mutant transcript. If we put this nonsense allele in the right genetic context, that is, if we delete a floxed allele, so remember, if you're homozygous for this, you're lethal, so we have to use a conditional knockout model where we inactivate a functional but floxed allele for NF1, and we do this in Schwann cell precursor cells using a DHH-CRE, what happens is we get tumors that arise along the spinal cord, and you see this classic kind of shape where these neurofibroma tumors start impinging upon the spinal cord. Our mice around 4 months of age start showing hind limb paralysis, and so you can start seeing that these animals are developing problems. If you look at EM, you see, or if you look at the sciatic nerve, you see swelling. You see myelination defects in the EMs for these nerves. So this is a great tumor model. We're now using nonsense suppression drugs. We've screened some mouse embryonic fibroblasts from these lines. If you knock out NF1, you see increased phospho-ERK activity, so it's part of the RASopathy. It's a RAS pathway inhibitor. If you delete NF1, you see increased RAS. So here's a homozygous, so we have increased RAS activity. The homozygous stop mutations, these MEFs have elevated RAS activity as well, and we can treat this with a drug combination of gentamicin, which serves as an aminoglycoside to allow read-through, and amlexinox, which suppresses the nonsense-mediated decay, that then allows us to restore or rescue partial activity in our cell lines. So now we have our mouse clinical trials have been underway, in which we've taken this same genetic combination, and we're trying to a treatment protocol. We let tumors develop. As I said, they start showing up around 4 months of age. Once an animal shows the presence of a tumor, it goes on the drug for 8 weeks, and then we rescan and look at activity. We also have a prevention protocol where as soon as the animals are weaned, we put them on the drug. And the prevention appears to, we're still early on with collecting data, the prevention may be working. The treatment trial, that's not working. So we're not restoring activity. I'm fairly confident in that study. Well, we also made a point mutation. So we had patients that had the spinal NF. And so we created, and so 2 unrelated patients, 2 unrelated families had the exact same mutation that had spinal NF. We made a mouse with this. Much to our disappointment, mice lived for nearly the same age. You know, they're out 2.5 years. No evidence whatsoever of any spinal tumors. So this particular mutation, if you look by Western blots, the homozygous animals appear to have slightly less neurofibromin. So we think this is a hypomorphic allele, but for the mouse does not recapitulate the phenotype that we see in humans. That so right now this is we go to prove this in a animal model. It's just an association right now, but we don't see these tumors. So I've pointed this out because we now have multiple mutations that we're interested in. This is a very large protein. We're now starting, we can't make, as I said, there's over 3,000 mutations so far. We're not making 3,000 mouse models of this. But we are selecting a few mutations that represent different missense mutations along with phenotypes. We have some where patients have dermal tumors, but they don't appear to get plexiform tumors. We have others that have the plexiform tumors but don't appear to get the dermals. So we're trying to figure out what are the functional domains, and is this somehow leading to interacting partners that could be targeted as downstream signaling pathways, or can we target the partners to regulate the loss of neurofibromin activity? Those first 2 mice that we made, one of them actually went fairly quickly. It took us about 9 months to make it. The other one took close to 2 years. And as I mentioned earlier, that was about average. 2 different projects, so it takes about a year per project. We now have these new mutations. We have multiple mutations, and we've made mice. We already have 4 on the ground in the last 7 months since we started working on these, or about 9 months. So we have 4 new mutations were made very rapidly using the CRISPR technology. We also have a new rat model that's on the ground that we're working with. We're also working with a group that just came to UAB to make a pig model. So with CRISPRs, we can more quickly make these. It's cheaper, and so we're now doing this at a much more rapid pace. The other thing we can do is make our cell line models using CRISPR-Cas much more quickly as well. And so what I point out here is we're making induced pluripotent stem cells for many of these patients. We're also taking a single cell line, so we have an isogenic line, and introducing the different mutations in that line, so we can really test, what is in an isogenic background, what are the consequences of these individual mutations that we're trying to study. We've created an NF1 deficient line so we can reintroduce the cDNA and also do some rescue type experiments with a single allele. For those of you that work with cell lines, be aware there's often duplication. So the HEC293 cells that's commonly used for drug screening studies is happens to have a duplication, and there's 4 different alleles for NF1, and so you have to be careful about sequencing and make sure you knock out all the different alleles, but this is fairly easy to do with a CRISPR-Cas system. We're also, there is kind of one hotspot mutation, so there's a patient that has a cryptic splice site. There have been 37 reported cases of this, and now what's known as exon 10. For the NF1 gene, and so one of the things we wanted to do is create a cell culture model for this in human cells and really validate, if we somehow come up with a therapy, if we can do antisense oligotherapy to suppress the aberrant splice site, this cryptic splice site, the mRNA will still have this mutation in it. So we had to test that in vitro, and now we're recapitulating this in cell lines to find out, well, we've proven that mutation, that if we make a full-length cDNA or full-length transcript, it does appear to function normally. Now we can go back and start modeling, well, how can we do antisense oligos to suppress this? So that's going to be great for doing that. So now we're using CRISPR in multiple strains of animals. We're using it in different species. We have a group at UAB who was just recruited about a year ago, Joseph Techter's group. This is going to be probably one of the clear clinical uses is xenotransplantation. So the ability to use pig organs, and Joe Techter's group is planning on kidney transplant, and they intend to be in humans in about a year from now. The key with the CRISPRs is we can now go back and knock out all of these bad viruses that you have to worry about by using another species, and so there's about 40 of them or so. You can go back and knock out all the individual pig viral components using guide RNAs to target those genes, and then you end up with a donor line that you can now produce kidneys that can then be transplanted into humans, so you don't have to worry about those additional problems. So a couple other uses of CRISPR-Cas9. We now have a dead Cas9, so you can inactivate the region that cuts the DNA, and you can also then tether that Cas9 protein to other effectors. So this has been done by tethering it to transcriptional activators and repressors. You're thinking, well, what use is this? Well, now we can go in and if you can put in a Cas9, you can target to a promoter region of a gene, for example, and carry along an activator or repressor unit so now you can dial up expression of genes selectively. And in fact, you can not only so this is first demonstrated with repression, you can turn down genes using a crab-tethered dead Cas9, using a VP16 transcriptional activator fusion protein to Cas9. You can upregulate gene expression. You can also do things like methylation, change the methylation status, or things like P300. You can do acetylation. So these are all these adapter molecules. You can now do very interesting things with the CRISPR-Cas system. You can tether it to, well, I'll come back to that in just a second. The other thing you can do is, many of you may have heard of optogenetics. So this is a way of activating in cell type, so do inducible expression by shining a light onto a tissue of interest, and you can activate in vivo the Cas9 system. And so this group has actually split Cas9 into 2 separate proteins, tethered these to made fusion proteins that are only activated, they will come together in the presence of blue light. They will then reconstitute the Cas9, which then allows you to bring in and get CRISPR activity. So just a very elegant way of regulating expression. There is a guide, there's a GFP tagged Cas9 that's out there that's being used for live cell imaging, so you can tag telomeres, you could look at particular regions. I don't think this sensitivity is enough here yet to replace what you normally do in the clinic, but you're getting close for screening. There's a whole host of Cas-like proteins, many of which have RNA activity, so these are just a whole new family that's going to be applying. Gene drive has been a concern, so we're now modifying gene organisms to maybe modify mosquitoes so you could release them into the wild such that the gene drive, if they have a Cas9 cassette, they'll pass on a mutation. And so the mutant mosquitoes really have a resistance gene to it that affects the parasites that carry malaria. So you can make mosquitoes that always pass on that mutation, and that's been a concern. And it kind of hit the brakes a few years ago about release, but DARPA just yesterday appropriated $65 million to do this for mosquito eradication, for treating malaria. Clinical applications are clear for this. So can we use the Cas9 to deliver genes directly to tissues? So if the liver often takes up things, so first pass, so can you deliver a Cas9 therapeutic agent to do gene editing in the liver? Or can you remove cells from the patient, do the editing, and then reintroduce those back into the patient? You've probably seen many slides like this, so making induced pluripotent stem cells, reprogram those, and then do things to do personalized medicine in the cell culture model to then tease apart pathways and then hopefully create the edited or correct the bad version of a gene, push these iPS cells to a cell type of interest, and then reintroduce those in the patient where it's possible. It's clearly going to happen probably first for things like where you can pull out these hematic stem cells or pluripotent stem cells, so treating sickle cell disease, probably one of the early successes. There's already been a couple of proof of principle experiments that have been demonstrated using this. The other area for the clinical applications is going to be things like chimeric T cells. So taking out these CAR T cells, which you can isolate T cells from a patient, introduce a transgene that then allows this to become more active and target tumor cells. So the CAR T cells are going to clearly be much more active in the coming years, and there's going to be clinical trials using these. And in fact, the first June of 2016, the Recombinant DNA Advisory Committee approved this. One of the things you need to do in the CAR T cells, if you mutate the PD-1 gene, that allows the CAR T cells to be more active, and so this is another improvement or kind of next iteration of the CAR T cells. Last week, FDA panel approved the use of these CRISPR-modified CAR T cells for the treatment of acute lymphocytic leukemia. So these things are happening. Clinical trials are rapidly coming on board for CRISPR-edited things. Another disorder by replacing this FAH gene in hepatocytes, and this is using an adenovirus that will deliver the Cas9 protein to in vivo regulate this. There are, treating viruses is another example of, that's what these Cas9-like proteins do best, is they fight off viruses. So treatment of HIV, there's clinical trials that are planned. And I'm running out of time, so I'll hurry up. There's one company that's already looking at hemophilia, Hurler syndrome, and Hunter syndrome. Now, this is a company that clearly has interest in the zinc-fingered nucleases. As I said, Cas9, they're simply nucleases. So what they do is they cut DNA and they do gene editing. There's an interesting trial that is being initiated for looking at HPV by using a Cas9 gel, or a gel that will deliver the CRISPR-Cas9 machinery to help fight off HPV. Huge number of other viral clinical trials that are applications are starting to go in. Here's a company that has all these different disorders that are already in their pipeline for CRISPR-mediated gene therapies. So targeting the eye is also another area which may be highly useful for CRISPRs, and that is because with the mainly because of the immune barrier for the eye, you'll be able to correct genetic mutations and restore sight. So I've given you a lot of data, a lot of examples of where we're going with CRISPRs. Here's kind of a summary of where we're going to see in the basic research laboratories. Probably this rapid testing of variants of unknown significance is going to be very important to clinical groups like this. So really proof of principle, and you can do this in cell lines and in vivo fairly rapidly with the CRISPR. I talked about all these different gene therapies and organ transplants, the antivirals. That's going to be probably the first uses. The low-hanging fruits are going to be enzymatic activity. So can we restore partial function partial gene correction. Those are going to be the disorders that work first. It's not going to be a key CNS neurofilament or something like that that's treated, because that's going to be too late for the patients. But some of these things where we can restore enzyme activity are going to be key. So lastly, I'd like to acknowledge my group, the Transgenic Core Facility, all the people who have made these animal models, and then close collaboration with the UAB Neurofibromatosis Program with Bruce Korf and Deanne Wallace in drug screening. And thank you for your attention.
Wow, thanks, Bob. That was great. Really gave us a great perspective on where this is going. I think we have time for maybe one or possibly two questions. Please, one of you, please.
Ask, or it's only one question. Right. So I think the better the animal model, the more impressive the data. So in many cases, if you go back and look at the hemophilia story or the hemoglobinopathies that have been studied, many of those genes have been completely humanized in the mouse. So in fact, the preclinical studies with them, it's actually the human gene in vivo that's been tested for the CRISPR activity, and then demonstrating that you can pull out from the patient the immune cells or the stem cells, do the treatment, and it's high activity and zero off target. So I think it's always going to be an issue in the back of your mind is what happens for these off target effects. Do we create a bigger problem than we have already? So I think that's where the bar was set really high because of the early adenoviral gene therapy studies. I think now the FDA is recognizing, okay, well, it's better to do something, so assume a little risk, but as long as you can manage that risk and demonstrate that it is exceptionally low, then you're going to get the go-ahead, I think.
So, fascinating talk. In the early days of gene modification and stuff, Europe got extremely exercised about changing sequences in animals and food and all this sort of stuff. And I can imagine this would really light them up. Can you say anything about how this is going in the sort of general European community?
Yeah, so that's an interesting thought, and they are much more risk averse than I would say people in the U.S. The risk aversion in China is very low, and some other areas. But I would say that I was a little bit surprised by I haven't get a chance to talk about, we've done some interesting things with swapping mitochondrial background with nuclear genome. The whole 3-parent babies that have been approved in Europe. And in fact, those originally limited to males only, but I think they've since released that. So the concern about introducing something into the human germline, it has been surprisingly not as concerned. Worldwide, there are strict restrictions. I didn't show a couple of the slides about modifying the human genome. So these are all going to be applications, but clearly you can't be modifying the genome that's going to be passed on. So I think time will tell. It's like anything. There's going to have to be some initial trials, and then we get more comfortable with the technology once there's a demonstration of very low risk. So, all right, thanks.
Okay, thank you, Bob. All right, our next